Good afternoon, everyone, and thank you for joining us today. My name is Katy Stubbs, and I lead the public engagement team here at Alzheimer's Research UK. It's my pleasure to welcome you to the 8th event in our Lab Notes online series. Today, we are hearing from our Thames Valley Research Network, who are taking us behind the scenes in their labs in Reading and Oxford. They've been filmed in their labs as they demonstrate the different techniques and experiments they use to make new discoveries about the diseases that cause dementia. They're then joining us live for the Q&A, so please do submit questions throughout the event, as we'll have lots of time later on to answer them. This event is being recorded and will be added to our website and YouTube channel early next week. If you've missed previous events, you can also watch any of them back in your own time. The videos that we're showing are already subtitled, but if you would like subtitles for the rest of the event, you can turn those on and off using the CC button at the bottom of your Zoom window. So before we get going, we've got a couple of poll questions for you. And these questions are a useful way for us to understand who's come along today. So why you might have come along, what your experience of dementia might be, how you might rate your current knowledge, and whether you've attended one of our events before. So hopefully those questions will appear. There they are. And we'll just pause for a minute or two to let you answer those. So if you've come before, you've probably answered these questions before, but they are really useful for us to just have a good understanding of the people we've got joining us and to make sure we are tailoring these events well. So hopefully if we've had enough people fill out the poll now, we can take a little look at the results. So people are you're rating your knowledge of dementia research around average. Hopefully by the end of today, you'll feel that has improved a bit. And we've got a mix of people reasons for coming along today. So I think the biggest one's actually people's work is related to dementia. And then second after that is that people are just interested in finding out more, or a friend or family member has dementia. And we've got almost a 50-50 split of regulars and newcomers. So welcome to you all. It's absolutely fabulous to have you join us as we share what's happening in dementia research. So the plan for today is first some quick updates from me before Dr. Mark Dallas will talk about the work of the Thames Valley Network, whose researchers will feature in today's event. We'll then have an artistic interlude with a poem from Jane Thomas before moving to the researchers taking us behind the scenes in the lab. We'll round off the event with a Q&A session, and we'll try to answer as many questions as possible in the time that we have. Many of you submitted questions in advance, and you can also ask questions during the event. If you'd like to ask a question, just look for the Q&A button at the bottom of your Zoom window, which will bring up the Q&A box where you can type your question in. So first up, I'm just going to share some slides and some exciting news from the world of dementia research. There we go. So hopefully you can see those now. So if you were watching the news at all last week, you might have seen some really big news about a potential new drug. So last week, the FDA announced that they have approved a drug called aducanumab for the treatment of Alzheimer's disease. And this is a landmark ruling, as it is the first new Alzheimer's drug in nearly 20 years, and the very first that works by tackling the disease itself rather than only treating the symptoms. The drug aducanumab is an antibody that is designed to target amyloid, a protein that builds up in the brains of people with Alzheimer's at an early stage of the disease process. It's not in a tablet or a pill form, but is provided in a blood infusion once every 4 weeks. In 2 phase 3 clinical trials, aducanumab was shown to reduce amyloid buildup in the brains of people with mild cognitive impairment or mild Alzheimer's disease. However, only in 1 of the 2 trials did aducanumab slow the decline in memory and thinking skills and in people's ability to carry out daily activities. Despite this mixed evidence, the makers, Biogen, took this as a positive sign that the treatment is effective, and they filed for approval in the US, the EU, and in the UK. So the FDA and the US are the first to make their decision, and it has caused some debate. The FDA believed that the reduction in amyloid is reasonably likely to result in clinical benefit, and so decided that the evidence in the application met the standard for what they call their accelerated approval. As part of this approval, they've said that the makers Biogen will need to carry out a new clinical trial to show that aducanumab does indeed have benefits for the symptoms of Alzheimer's disease. So here in the UK, we are now waiting for the decision from our own regulators, the MHRA, and whether they will also approve the drug. If they do, then NICE will have to weigh the benefits of the drug against its cost before deciding if it would be provided on the NHS. We expect a decision from the MHRA in the autumn, and then a decision from NICE probably about 10 months after that. This means that if it gets through these stages, doctors could be able to prescribe this drug from mid-2022 at the earliest. So here in the UK at the moment, we have to wait and see what happens. And at Alzheimer's Research UK, we've been working in recent years through our Dementia Access Task Force to help prepare the NHS for the delivery of any new treatments. Over the past year, we've seen how the MHRA have sped up their review of vaccines and treatments for COVID-19 without compromising on the quality of their process. So after last week's announcement, we wrote to the Health Secretary, Matt Hancock, urging him to intervene to accelerate the MHRA's review of aducanumab. People with Alzheimer's disease and their families have been waiting a very long time for news like this, and so they should be given answers they need as quickly as possible. The approval of aducanumab in the US doesn't mean that this is job done for dementia research either. The FDA's decision is seen by many as a positive step, and we hope to see more treatment for Alzheimer's and all the other types of dementia show positive benefits for patients and get approved in the coming years. Right now, there are over 150 clinical trials of 126 potential drugs for Alzheimer's currently taking place, and there are many more for other types of dementia too. And researchers like those you'll hear from today are working to identify potential treatment avenues to explore and develop these much needed treatments. And kind of in hand with that, you may have heard in our previous Lab Notes events about our petition, in which we called on the government to honour their election promise to double dementia research funding. With over 51,000 signatures in support, yesterday we delivered our petition to the Prime Minister. Behind each signature is a story of how dementia has affected people's lives and the urgent need for research to improve diagnosis and treatment of the condition. Pictured here is Olive Munro posting her letter to the Prime Minister that tells her story. Olive witnessed the heartbreak of dementia in her family, with 3 aunts dying with Alzheimer's and her cousin now living in a home. She herself is living with vascular dementia and believes that research can make vital breakthroughs, which will mean her grandchildren will not be affected in the same way. So a huge thank you to everyone who supported our petition, and we hope to bring you more news about progress in this area soon. So today we're hearing from researchers in our Thames Valley Research Network, and it's my pleasure to hand over to Dr. Mark Dallas, who leads this network, so we can hear from him about what the network is about and what they've been getting up to. So without further ado, it's my pleasure to hand over to you, Mark.
So welcome everybody, and thank you very much for that introduction, Katie. It's my pleasure to be kind of hosting this session on behalf of the AR UK Thames Valley Network, and in my role as the academic coordinator. So by way of introduction, my name is Dr. Mark Dallas. I'm an associate professor in cellular neuroscience based at the University of Reading. And so by way of introduction, I'm going to share a few slides just to give you a little bit more information on what the network is, what we try to achieve through our researchers, and then we'll go on and showcase some of the work that we've been doing through the video and also our creative interlude, as Katie mentioned. So I'm going to share my screen with you, and that'll have some slides for you. So hopefully you can all see the screen.
Yep, they're up. Thanks, Mark.
Perfect, thank you. So again, what is the network? So our network brings together a community of research scientists from 3 different institutes. So here at the University of Reading, we're joined by colleagues in the University of Oxford and also Oxford Brookes University. And really that's the key to the power of the network, collaboration and bringing experts from different fields together to tackle dementia. So here's some mugshots of all of our kind of collaborators and scientists. And I want to draw your attention to one particular person, and they aren't a scientist. So this is Lorraine, our administrator. She keeps us, our scientists, on track and allows us to carry out the science in a very efficient manner. So the actual network itself receives funding from Alzheimer's Research UK, and we use that to support pilot research, so starting to foster some new collaborations into new avenues, as Katy mentioned. We also provide funds to support travel. So while things have been restricted recently, in normal times, our scientists go off and share their research on the global arena to showcase the research that they've been doing and international collaborations to build a global presence in terms of tackling dementia. Katy's already mentioned that research is key. And again, it's us about tackling dementia from different aspects that will provide the answers that we really hope to provide to those living with dementia. So this is how we imagine our research network. At the top here, we have medicines. So we have researchers that are looking at medicine use in those living with dementia, but we've also got people trying to work out and make new medicines. Katy mentioned the FDA approval last week. Again, our scientists are hard at work to produce the next medicine that could hopefully be approved here in the UK as well as in the US. We've also got clinical colleagues who are working hard to look at those living with dementia through imaging studies to try and monitor the changes that we see in brain function and brain anatomy, to see if we can detect and diagnose things earlier, or see if we can intervene to try to modify some of those changes that occur. We have a whole host of researchers that are looking at basic neurophysiology, trying to understand the cells within the brain, how they communicate, and what goes wrong in diseases such as dementia. By understanding the basics of how the brain works, we hope to then be able to build a better picture of what goes wrong and where new medicines and treatments could be of benefit to those living with dementia. And then finally, we think about pathology. So looking at tissue from those that have been living with dementia. What's the differences? Can we spot key differences in order for us to diagnose and detect dementia at a much earlier time point than we do currently? And again, that's something that our research community is hard at work in doing, in trying to find those biomarkers that will be key to getting that early diagnosis and earlier treatment, and hopefully better outcomes for those living with dementia. So we're going to hear this afternoon from 3 of our researchers, and they live and work in institutes across the network themselves. Our first port of call is the University of Reading, and you'll hear in the video later on this afternoon from Dr. Francesco Tamanighi, and he tells you a little bit about his work in the laboratory. We then head to the University of Oxford, and we have 2 researchers there that go into their lab and share their work in the laboratory with you. And that'll provide insight into some of the equipment they use and some of the experiments they are carrying out to tackle dementia. But it's not just all about research. One of the things that we are keen to do as a network is try to make that science accessible. And one of the ways we do that is collaborating with artists. So here on screen, we've got some images that were provided by a collaborative award in collaboration with Dr. Lizzie Burns. So by doing these colorful images, we make brain science, which is very complex and challenging even for scientists like myself, to try and explain what goes wrong to a range of audiences like yourselves and even those younger years at school. And I'm delighted to say that for today, we've actually collaborated with a poet. And she has actually commissioned her own poem which gives a little insight into the research that goes on within our network. So I'm delighted that Jane Thomas has recorded her poem for us, and I'll let her introduce it, and we're going to hear from her now.
Delight to be part of such an inspirational and amazing day. I am Jane. I'm a poet based in Oxford. I am just finishing a collection which is based around Alzheimer's and dementia, partly based on some of my own lived experience with my own dad. I'm here today to read my poem, specially commissioned for this event, and it is called Your Brain. Your brain was like the old Royal Mail train, ever sorting messages in motion. It left at 5 a.m. every day, stuffed with brown bills, birthday cards, love letters, north to south, the old-fashioned way. Exact sorting at dark speed as it swayed, gliding through Crewe, Milton Keynes, and finally into the tunnel at Waterloo, working at full tilt until it didn't. Until it turned back to the 70s and the winter of our great discontent, when the dustbin men went on strike and the porters and grave diggers too, and all the black rubbish bags piled up in the squares and coffins waited to be filled, and cards and love letters went astray as mail trains rusted or went the wrong way. Thank you.
Thank you, Jane, for sharing those inspirational words. And again, it breaks down dementia and makes it accessible. And that's using her lived experience of dementia, and also in discussions with scientists here within our network. So now we're going to move on to the behind the scenes look. And again, we're sharing some stories from researchers across our network, and they are going to be showing you into their laboratories. A little bit of the science, showing the equipment and some of the people they work with on a daily basis to tackle dementia. So I hope you enjoy the video and it provides you some information. And here's the video for you to watch now.
Hi, my name is Francesco Tamagnini and I work here at the University of Reading School of Pharmacy. I am a member of the Alzheimer's Research UK network for the Thames Valley. And today I will talk to you a little bit of what we do in our lab here at Reading. My group focuses on characterizing the electrical alterations and the structural alterations in the brain of mouse models which express proteins which are found in the brain of people with dementia. So we do have different approaches. We use genetically modified mice which express these proteins, or we take these proteins and we add them in the culture medium of petri dishes which contain these nerve cells, or we take sections of the brain of perfectly normal animals, keep these sections alive in a solution called artificial cerebrospinal fluid, and add these proteins to that solution so that the brain slices are exposed to it for a certain amount of time. And then what we do is to place these brain slices into a machine called electrophysiology rig, which in this case is also implemented with a specific very powerful microscope called a two-photon microscope, and that will allow us to measure the alteration in the electrical activity of single nerve cells into a complex network after they are exposed to proteins which are associated with Alzheimer's disease and dementia. Here you can see these faint, but it's actually quite crispy considering that the sample has not been stained in any way. These pear-shaped cell bodies, here you can see one a little bit better, yeah. These are neuronal bodies. If you squint, you can even see the processes going down. So all these neuronal cell bodies are in the part of the brain, the hippocampus, which is involved in the encoding of new memory. So what we do is to fill up these cell bodies with a probe, a glass pipette filled with a solution that resembles the content of the neuron. And on top of that, we have a fluorescent substance, which in a second I will show you how it will enable us to reconstruct the structure of the neuron within this complex native matrix. At the same time, this micropipette is connected to a complex electrical system, which will enable us to visualize the electrical activity of this neuron live on our screen. What you see here are some pre-recorded, so to speak, wobbly lines. So what are these wobbly lines? If you get close enough to the cell, right, and then the plasma membrane of the cell gets attached to the glass pipette, and then you break the piece of plasma membrane that there is between the pipette and the cell, so the inside of the pipette is in contact with the inside of the neuron, then you will be able to measure the electrical activity of that neuron. This is a specific technique that is called voltage clamp, which allow us to measure the electrical currents that flow through the neuron. So you see here that you have these spiky-shaped electrical currents. What are they? And why do we care about that? Well, we care about this because we do know that these currents that we record from one cell are evoked by the neurotransmitter that is secreted by the presynaptic cell. So when the neurotransmitter binds the postsynaptic cell AMPA receptor, that's how it's called, sodium enters the AMPA receptor, generating an electrical current that we can measure. And what can we measure? We can measure its amplitude. We can measure the frequency of the events. As you can see, there are many events. We can measure, how fast the current is generated and also how fast the current disappears. And as a matter of fact, what we did observe in this model of tau pathology is that these currents are altered in presence of the tau protein. Now, the next step of the project, which will be the object of the next grant application, is to understand why tau proteins lead to the alteration of these waveforms. So, as I was explaining, this is the chamber in which we have placed the brain slice. You can't see it because right now it's sandwiched between the bottom of the plate and the powerful objective of the microscope. So it's kind of a busy scene, yeah? You do have the slice, you do have this probe, which is the temperature probe. You do have this probe, which is the ground, the electrical ground of the electrophysiology probe. You have this probe, which is connected with a glass micropipette filled with internal solution. This is a preamplifier, which then is connected with an amplifier. And here you've got the whole machinery of the two-photon microscope. So in this rig in particular, we can basically measure two main things: the electrical properties of the plasma membrane of the neuron, and also image the structure of the neuron as we have filled it with the internal solution, which contain, and maybe you can see it here, it's a little bit yellowy, a fluorescent probe, which is called Alexa Fluor 488 probe. This is particularly good because it's a hydrazide, which means that it remains trapped inside of the neuron once you have injected it inside. So before we started filming, I've actually prepared an experiment. So this image will not be an image that you see in a camera, will be a reconstructed image, because what my laser will be doing is to scan the sample by exciting it and recording the light that comes out of it. Once we are done with our acquisition, we end up with a stack of images. So you can see here that there is the pipette, And as I move my cursor, each one of these is a different focal plane, yeah? And then as I move down my focal plane, you can see all the cell body of the cell, and you can see the single processes moving throughout the thickness of the different layers. So I can adjust the brightness to make it a little bit brighter. Now that is one level of magnification. Potentially you can go with a much higher magnification. So this is an example of a magnified 4 times neuronal body. So if before you've seen the cell shape and the pipette, now if you go and focus or you actually zoom your scanner onto a process, you can see these are the single branches of the neuron. And if you look carefully, you can see that these branches have these little tips and dots like little spines, and they're called exactly that, spines. And these are the functional compartments where new memories are presumably shaped. And in Alzheimer's disease and in tau pathology, we do have alteration in the structure of these spines, which mirror and to some extent are causative of the electrical changes. So the beauty of this machine is that will allow us to monitor both the structure and the function of the neuron at a fairly high resolution. As we were seeing at the microscope, at the rig, with that machine we can get 2 kinds of data. Yeah, we can get the electrical activity of the brain, or in this case of the brain slice, related to the network, yeah? So how cells communicate with each other. So these events here called synaptic events or excitatory postsynaptic currents in this case. But also another kind of data that we can get are, the intrinsic electrical properties of the plasma membrane. So for example, with this technique called current clamp, patch clamp, we can monitor the potential of the cell while we inject current steps. So if we inject a positive current step, we can drive the cell to fire these these electrical spike events, which are typical of nerve cells or of excitable cells in our body. These events are called action potentials, and they work a little bit like an analog digital board. Yeah, they convert. In a way, we are robots which work with these digital events, right? It's the way the cell communicates. Or the information travels across the cell. And also we can measure the currents, the electrical currents that are generated on the plasma membrane to generate the action potentials. So specifically an inward current of sodium ions, these electrically charged particles of sodium known as sodium ions, and the outward current of potassium ions, these electrically charged particles of potassium, which are both needed for the generation of the action potential. And of course, we can compare the properties of all these waveforms between normal mice and mice which have a genetic mutation to express, like in our case, a form of tau that is associated with Alzheimer's disease. And as I was saying, on top of the functional stuff, we can do all sort of structural evaluation directly onto the brain slice or the cell within the brain slice, so measure the number and the shape of synaptic spines in our neurons. This is just an example of a 1x image as we were seeing at the microscope, and a 4x image. So this is not an optical zoom. This is actually a scanning zoom. Thanks to various techniques of manipulation of the image, we can actually resolve very fine structures. Okay, so our day here at the University of Reading School of Pharmacy is over. We've been through, quite a few aspects of what we do here. I hope I convinced you that our techniques are somehow useful to understand what goes wrong in the brain exposed to these proteins, which we know are involved to a certain extent in the onset of dementia. But some could ask, well, then so what? Once you have seen that these brain slices have unaltered synaptic activity or unaltered electrical activity, when they are exposed to tau protein or amyloid protein, or they've got weird processes coming out which are not looking exactly as they should, then what? Well, that's actually a very good question, but I think that the essential part of this research is that it allows us to understand what goes wrong and what molecular target is responsible for these changes. It can be a protein, it can be a protein channel in the plasma membrane, it can be even the composition in the phospholipids of the plasma membrane, those fatty sheets that form, the perimeter of the plasma membrane. And once we better understand what is the molecular target that is responsible for these changes, then we have a target for example, a new drug or a new non-pharmacological intervention. Basically, what I'm saying is that for you to be able to develop novel, effective therapeutic strategies for this disorder that right now doesn't have a cure, you need to be able to identify new targets. And the way we do that here is to try to identify targets that are linked to the alteration in the electrical properties and activity of nerve cells.
Hello, my name is Fran and I'm a member of the Alzheimer's Research UK Thames Valley Network. I work as a scientist here at the AAUK Oxford Drug Discovery Institute, for short, the ODDI, aiming to defeat dementia. I'm sitting outside the NDM research building, which accommodates the drug discovery's facilities. But let's go inside and see what's going on in there. We will go all the way up to the top floor. The ODDI is one of 3 AAUK-funded institutions. And our group here in Oxford is a team of experts in different fields, such as chemistry and biology, combining both academic and industry knowledge. Together, we want to tackle Alzheimer's disease along with other forms of neurodegenerative diseases and discover novel, effective therapeutics. We are sharing a large office and lab space with other groups, which enables us to communicate and discuss our science and help each other. I will now introduce you to some of our scientists that work on AAUK-funded dementia research. All of us are using and sharing a wide range of equipment in our lab. We have all been working throughout the pandemic under safety measures to ensure our research progresses. The journey of drug discovery here in the ODDI starts in the chemistry lab, where compounds are made from tiny molecules. Personally, it always reminds me of cooking, mixing ingredients that react together and form an end product. I want to introduce you to one of our hardworking chemists, Trifon.
Hello, my name is Trifon and I work in drug discovery. I am making a compound that aims to prevent processes in brain cells that lead to the development of Alzheimer's disease. The compound stops the cells from producing an unwanted complex of proteins that, if left alone, would lead to Alzheimer's. I make the compounds by combining different reagents we have in the lab. Then I purify the final product to test in our experiments.
So we have the compounds made by the chemist, and they prepare them for us in a concentrated stock solution. But what happens now? Please meet my colleague James to learn more about how we use these stock solutions of these compounds.
Hi, my name is James. I'm a biochemist and I am testing new compounds sent to us by the chemistry team to see if they alter the activity of our target enzymes. A target enzyme is one that we have identified as playing a role in causing dementia, and we hope by altering its activity inside the body we can help treat the disease. When we receive the chemicals, we dispense them into these plastic plates with tiny wells for each chemical. To dispense the chemicals for testing, we use this machine here called an echo. This machine takes our highly concentrated stocks of compounds and uses sound waves to ping tiny droplets of drug from our source plate into the wells of our experimental plates. The robot arm attached to the echo can be programmed to automatically insert and remove the plates. This allows us to precisely dispense thousands of compounds much more quickly and accurately than we could do by hand. The experimental plates are then ready for the enzyme to be added or can be stored in environmentally controlled pods until we need them.
Please meet Grace. Grace is not only committed to ARUK-funded science, but she is also a member of the ARUK fundraising team here in Oxford, helping to support dementia research. Grace will tell you more about the effects of compounds in organisms, more particular in cells, the smallest functional units an organism is made of.
Hello, my name is Grace and I'm a research assistant here at the ODDI. Currently, I'm researching the role of inflammation in the brain and how this can be prevented with the new drugs made by medicinal chemists like Tryphon. To look at the strength of our new compounds, we first do this in cells that are grown in a flask. Here you can see the layer of cells growing under the microscope. From the flask, we transfer the cells into a plate and add each new compound to different wells along with a stain. This makes the cells fluoresce brightly, so we can see different parts of the cells clearly when we image them. And now for the fun part. This is our Opera Phoenix. It is a specialised piece of equipment that allows us to take images of the cells in great detail and see the effects the drugs are having. For example, using the staining, we can see the appearance of these bright specks inside the cells, which are signs of inflammation. This one is important because it doesn't have any specks. Using this technique, we can look for new drugs that prevent the appearance of these specks. The aim is that these can be taken further as new drugs for Alzheimer's disease.
And now it's my turn to tell you about my research. This equipment, called an octet, is used by myself a lot. I'm investigating a protein that is involved in the progression of Parkinson's disease. Ideally, we want to inhibit this protein with inhibitor compounds to stop the disease progression. I'm using these sensors that will be coated with this protein, and these sensors will be dipped into inhibitor compounds. With this technique, we can optically detect interactions between target and compounds. We can measure how well these compounds bind to our target, which is a very important information concerning the effect of this compound as a potential treatment for patients. Hector is working in the darkroom lab, where light-sensitive experiments are taking place.
Hi, my name is Hector and I'm a research assistant here at the ODDI. Today, I'm using our Flipper Tetra instrument to evaluate the effects of some of our new compounds on a protein called SHIP1, which is involved in the progression of Alzheimer's disease. We are trying to find a drug that inhibits SHIP1 to hopefully help patients. With this machine, which is basically a very precise camera, we are able to detect phosphate as it is produced by SHIP1 and ultimately find compounds that inhibit its activity. So I'm actually looking at these two curves here. This one represents the reactions as it happens without an inhibitor, and this one represents the reaction with an inhibitor. So we're looking at compounds where the reaction will look like this rather than like this. So this is the undisturbed reaction, and this is the one when we've added our compound, and we're looking basically for that reduction in signal. So here on the right, I got basically all the wells in the plate, and then I can see all the different curves, click on them and get the different curves that come out, so I can evaluate each of these represents a specific compound, and I can look, oh, this one didn't inhibit or another one inhibited basically. This one, reduction. This one a bit less of a reduction. And I'm looking here, there's a difference. These ones are active, this one's inactive. You can see here a trace which represents the amount of phosphate produced and how one of the compounds stops that reaction.
Joining us in the darkroom is Juliane. She's working with a type of brain cells called neurons. These can be grown in a dish. These neurons are very important for studying and understanding the disease progression of dementia.
Hi, I'm Juliane. I am using the microelectrode array, or MEA, to study the function of neurons in vitro. Neurons are the cells in the brain which commonly die in neurodegenerative diseases, such as Alzheimer's disease. Under normal conditions, neurons form spontaneous networks and communicate with each other by generating electrical signals. And with the MEA, we can measure this electrical activity in real time. Therefore, the neurons are cultured in a specific plate, which has a grid of small electrodes embedded in the culture surface to detect the electrical activity. This plate-wide activity map shows the neuronal activity across the plate, with each square representing a different well. If we take a closer look at the specific well, we can observe the neuronal activity in the form of so-called spikes and network bursts, which are key features of healthy and mature neuronal networks. This is a great system to characterize our neuronal cultures and to study disease-related mechanisms. We also use the system to test compounds in order to see whether they modulate neuronal activity and to screen compounds for safety and efficacy.
Special thanks to Chris. Without his coordination and help, the tour today would not have been possible. I hope this virtual lab tour gave you an impression of what our AAUK-funded scientists do and how the funding is being used to help to defeat dementia. We had a lot of fun making this video for you whilst showing our science and equipment. We are very proud to be part of AAUK-funded drug discovery.
Hello and welcome to the Lab of Molecular Neurodegeneration here at Oxford University. We're just going to have a look in the lab at the science that we do. Come on in and follow me. We're now going to have a look at Naroa's research. She actually investigates stem cells that we derive from controls and patients, and she produces neurons and astrocytes, which are glial cells that she's going to talk about, because neurodegenerative disease is not only about neurons, it's also about glial cells. So let's see what science she's doing today.
My project focuses on the role of glial cells, such as astrocytes and microglia in Parkinson's disease. Many people think that we only have neurons in the brain, but we also have these other cell types that are really important for the neuronal survival and the functioning of the brain. Now I'm gonna be feeding some of the astrocytes. These are over 80 days. They have been cultured for over 80 days, and they have been derived from stem cells that were derived from patients. So I need to feed them every 2-3 days, depending on how fast they are growing. Here, as you can see, I'm giving them some cell meat. And here we have a microscope because we can see the cells just with our eyes because they are really small. So we have to come here and on the screen check every day how they are looking, how they are growing. And I'm gonna show you now some really nice astrocytes, and these are over 80 days. Yeah, here you can see a really nice and big astrocyte with a lot of processes. And as you can see how they are interacting with other cells nearby. And these other cells are neuronal cells. And as you can see, they are smaller than the astrocytes before. And they have many thin processes to interact with the other cells. Cells are kept at 37 degrees in the incubator. Therefore, when we are looking at them in the microscope and they are room temperature, we have to keep them out here for as short a period of time as possible, because they like, as ourselves, they like to be nice and warm inside the incubator.
We're now going to have a look at Marta's research. She is really interested in lysosomal biology, which is the recycling bin of the cell. The cell also needs to recycle its materials and to take away anything that might be detrimental to it. And so Marta's really interested in looking at this, and she's going to show us how she does it in the lab.
So I'm using primary cultures to study neurodegenerative disorders, and I'm looking particularly at lysosomal pathways. And lysosomes are essentially the bins of the cells. And it's very important for neurons to be very efficient at recycling all their components to ensure that they transmit signals properly. And over time, and particularly in neurodegenerative disorders, this mechanism becomes impaired and dysfunctional. So we focus on ways and avenues to bring this pathway back into function, make it more efficient so that neurons can function properly as they should. We study neuronal cells, but also it's important to look at other types of cells that interact with neurons. And in this case, we're looking at astrocytes particularly, but it is also possible to look at other glial cells. And it's important to take in consideration the interactions between these types of cells and how they might influence lysosomal pathways and mechanisms, particularly their contribution to neurodegenerative disorders. So here in green we can see neuronal cells, and in pink we can see astrocytes. And it's particularly important to look at these two types of cells as they will interact in the brain environment, and they will both contribute to neurodegenerative disorders. And when we focus on lysosomal pathways specifically, it's important to consider their interaction. Even other types of glial cells, as they will contribute differently to the brain environment and therefore to neurodegenerative disorders as well. Now, looking at specifically lysosomal pathways, both neurons and astrocytes will contribute to disease and dysfunction.
After having a look at Marta's and Naroa's science, we're now going to have a look at a new method to detect tau pathology in Alzheimer's disease. So here we're looking at a control brain. We can see the nice neurons in this brain, and that there's very little to no pathology in the form of very tiny puncta that we've shown can detect early tau aggregates. However, if we look at advanced Alzheimer's disease brain, we can see that there's quite a lot of puncta, which are these little tiny dots that represent aggregates. If we look not too hardly, we can already see tau tangles, which are part of the neuropathological events necessary to actually diagnose Alzheimer's disease. However, the challenge was to try and detect this aggregation and this aberrant clumping very early on, so that when disease is just barely started, we can already find it in patients and use this detection technology to find drugs that will tackle the process early on, and so try to modify disease. And this is a patient at ROC stage 1-2, which means that disease is quite early on, and we can see that there's quite a lot of puncta, even if we don't find a lot of tangles as we scroll around, because it's early in the disease, so tangles are not abundant at this point. And this technique is actually really interesting because it can highlight this very early pathology and allow us to develop drugs in this way to tackle it. So here we have some human tissue, human brain samples, and you can probably follow the S-like structure, which is the hippocampus, in all of these brains. And what we have is a control patient, in Braak stage 1-2, which is really early Alzheimer's disease and advanced Alzheimer's disease. And this can take us back to the ARUK Orange campaign, where we can see that there is quite a lot of cell loss when we look at the tissue that is in the hippocampus and the brain cells that are MB0 versus Braak stage 6 or B6. So we can see that shrinkage and that loss that in the end is the size of an orange, the volume of an orange. ARUK funded this very important project in order to detect early tau aggregation. And tau is a protein involved in Alzheimer's disease, particularly in neurofibrillary tangles. And it's a requirement for the diagnosis of Alzheimer's to have tau pathology. And so what we can see between these brains is that the cell loss between B0 and B6 is dramatic. And we were very interested in detecting tau aggregation early on. So not only in patients where it's advanced, but in patients B1-2 or Braak stage 1-2, where disease has just set on in order to be able to tackle disease early. Thank you for joining us at the Lab of Molecular Neurodegeneration here at Oxford University. I hope you found it interesting, and we'll hope to see you soon.
Wonderful. Well, thank you to all the scientists that featured in those videos. I don't know about all of you, but I thought there was just so much to dig into there. So do start submitting any questions that you have, and we can really delve into it. And I'll just wait while some of those scientists that were featured come on screen, and we'll head into the Q&A session. So really, I'm really gonna struggle where to begin, but maybe I'll go to Nora, who was the last person to speak there, and you were showing this tau protein and talking about how it changes in Alzheimer's disease. And I just wonder if you could maybe tell us a little bit more about why this tau protein is important.
Absolutely. Well, we know, I'm sure everyone in this audience knows that neuropathology-wise, we have amyloid plaque, and that's what you've talked to us about, what aducanumab actually tackles. But there's also neurofibrillary tangles inside the neuron with a protein that starts disease within the neuron, which is tau, and actually correlates very well with all the traits of Alzheimer's. And it's just very difficult to tackle tau because it's intracellular as opposed to extracellular. So everything, if there's there's always barriers for us when we create a drug. First, it has to get into the brain, and then it has to get into the cells in the case of tau. And so that poses one of the complications when tackling it. And the other complication was that we couldn't really understand pathology very well without looking at intermediate aggregates. Aggregates come in many flavors. They're not only the big chunks that we imagine, the tangles and the plaque. If you think about Legos, that would be a massive Lego the size of this room, but tinier pieces of those Legos, like maybe 8 Legos stuck together, those are also quite damaging to the brain, be it tau or amyloid, because they can interfere with a lot of the cellular processes that neurons need for correct functioning.
Wonderful. That was a really good answer. And then I think we've had a question come through, so I'm going to stick with you, Nora, because I think you said you can answer this one. And it's Diana's asking that she understands that some brains that have been examined after death have been free of this amyloid protein. Is this something that you know about? Does this make sense in terms of what else we know about Alzheimer's disease? And then kind of related to that, how can we know if somebody has this amyloid protein in their brain?
Yes, they're both really good questions. So there's a really good correlation between tau and all traits, as I was saying. The traits with amyloid plaque, or the classical, how we classically defined amyloid plaque neuropathologically, was not as clear. There seems to be an increment with the disease, but there can be Alzheimer's patients where we evaluate the brain post-mortem, and if they're in Braak stages from 1 to 3, they can have quite little amyloid plaque there. And at the beginning, that was quite puzzling, because plaque recruits a lot of things in the brain, and it is quite like a very, very pathological feature. And it was very puzzling, but researchers have ever since established that what we were classically looking at was that big Lego that I was talking about, the size of this room, and that intermediate species correlate much better when we talk about amyloidosis in A beta and disease. And so if you look at the different pools, and if you have different bags of the different types of Legos, then that actually correlates very well with disease. And then how to image it? Well, we have an amyloid PET. And that's actually how they saw the reduction in the aducanumab study in amyloidosis. There's this radioactive tracing that people can get injected with that will stick to the amyloid in the brain, and it will light up the brain. And I think you had a very nice image at the beginning of the presentation. And it was shown that in very early Alzheimer's, there was a reduction to an extent of this amyloidosis.
Wonderful, thank you. So I think the next question, I'm going to come to you, Mark, because it's about the network as a whole. So somebody's asking, do you do any non-biological, so psychological or social research in the Thames Valley Network? And to what extent do you link with local mental health and community NHS trusts to answer important research questions? So I wonder if that's one you can handle for us.
Yeah, I think while the kind of mainstay of the research that we take here at the network is answering sort of biological questions, there is a place to investigate psychological issues. So we have colleagues here at the University of Reading that are looking at how indeed language can affect dementia diagnosis, and whether those that speak many languages indeed are offered preventative or protective measures against going on to develop dementia per se. And again, the key link here, again, thinking the mention of mental health, it's thinking about how we can tap into those individuals that are at risk and potentially try to monitor some of the changes. I think one of the key things, and researchers across the world understand, is there's commonalities in very many brain diseases, and it's trying to then couple those together to make it beneficial in terms of a medicine or a therapy in tackling dementia. So short answer is yes, we draw on all our expertise within the network, and that includes psychologists and local community outreaches in terms of mental health.
Wonderful, thank you, Mark. So my next question, I'm going to come to you, Franziska, and it's about some of the, I would say, quite amazing technology that you showed in your film that the Drug Discovery Institute uses. So there was the robotic arm that can move things with sound waves, and this just seems to be that technology is really, really important for your work. So, I mean, how new is some of this technology? Is it that actually we couldn't have done this sort of dementia research and developed treatments in this way even 10 years ago or before that? I don't know if you can speak about that.
Yeah, this is a good question. So I think it is a big advantage to have these robots we use because these compounds we use, they have to dispense in tiny little nanoliter, femtoliter droplets in order to use them for high throughput experiments. And without these robots, it would be not possible. And we can test so many compounds in one go. So it's very fast, our research, in comparison to if scientists have to pipette it all by their hands. Yeah, so absolutely, we are very dependent on this kind of equipment, and it's very state of the art. Yeah.
Wonderful, thank you. And thinking along the lines of equipment as well, so Francesca, you showed some really high-tech bits of kit that you use when you're trying to record from the brain, but you also talked, so you showed us quite a lot about what you can do, but I wonder if you can expand on some of the things you said at the end of your video about what some of your work might mean. So you can record these kind of electrical signals and see how they're changing in Alzheimer's disease, but what do you think that this kind of means? What's the findings that you're getting?
So much of the research in the last few years has been focusing on the hallmarks of Alzheimer's disease at a histopathological level, so amyloid beta and tau pathology, and that's perfectly fine. But in over 20 years, we've seen that not much advancement has been done in terms of clinical outcomes, right? So I think that kind of motivated the push for finding other kind of targets. So what I do is to work down the line of the causes, right? So the idea is that you have some initial event, whether it's amyloid beta or tau, and that alters the biochemistry. So some enzymes going on and, working in the brain, such as kinases and whatnot. But then at some point that needs to lead to the cell death and to the neuronal dysfunction, because the behavioral and cognitive alterations that we do see are caused by, altered electrical activity in the brain. So the idea is, what if I can tackle that down? If I can somehow understand what goes wrong between the amyloid beta and the tau pathology and the clinical outcome at a neuronal and functional level. Also because it has been observed in people with Alzheimer's disease that there is this higher risk of epilepsy and hyperexcitability. And people would think that there's a reduction in the electrical activity in the brain of people with Alzheimer's disease. Actually, what we do observe is quite the opposite. And so the idea is to try to find a potential target amongst those proteins in the plasma membrane, which are responsible for the generation of the electrical activity, such as ion channels or neurotransmitter receptors and whatnot. And even if we don't find a target in those, in the pursuit of this research, we will better understand what is the sequence of events, because right now we don't know it very well. We know that there is a starting point, an end point, and we do know something about what occurs in the middle, but we don't know the fine details of the functional alterations. And so my work in the last 10 years basically has been focused on this.
Fabulous, wonderful. And so your work focuses just on Alzheimer's disease, am I right? But I was wondering, do you think that there might be a kind of common thing that goes wrong in different types of dementia to do with something like electrical excitability of the neurons, and that might give some really important insights and kind of a common way, a similar way to treat lots of different diseases?
This is for me, yeah?
Yes, Francesca.
Yeah. So, no, absolutely, yes. I mean, if we learned something about these clinical trials, is that not necessarily the drugs don't work. Maybe our classification of dementia based mostly on clinical outcomes with some reference to the amyloid load and tau load probably is not so accurate. We need different ways of stratifying. Maybe we're dealing with different diseases in terms of ethiopathology with similar outcomes, and so that happens only by doing more and more research. So for example, what we did observe in terms of electrical activity is that while amyloid pathology tends to be associated to hyperexcitability, and that has been shown in different models of amyloid pathology, transgenic mouse models, starting with mucus work back in 2007. These little mice showed spontaneous seizure-like, epileptic-like behavior in the hippocampus. Tau pathology doesn't seem to be like that at all. Tau pathology is associated, well, in general, to reduce the excitability, but more specifically to a change in a protein called HCN channels, which are involved in the single cell encoding of network activity. I will not get into the technical details, but there's in my opinion, there's definitely something there, you know, to be dug out. Something very similar occurred years ago with breast cancer. Breast cancer was considered one disease. We do know that it's not one disease, it's many different stratified diseases. And the efficacy in the therapy against breast cancer derives exactly by the ability to differentiate the different pathways that you have in the different subkinds. I think with dementia, it's going to happen something similar.
Brilliant, thank you. And on a similar note, I'm going to come to you next, Franziska, because the Drug Discovery Institute is trying to find potential medicines and treatments for lots of different types of dementia. So I wonder, thinking not just of Alzheimer's disease, but maybe another type, can you talk to us about some of the work that's going on or some of the different types of dementia that you're trying to design new treatments for?
Yeah, so we are not only researching on Alzheimer's disease, but also Parkinson's disease. So these are the main two ones our focus is on. And yeah, so we tackle the disease from different angles. We have little sub-teams in our institute, and they are working on different angles of the diseases. We have the biology side, we have the chemistry side, we have protein biochemists. So we try to tackle both of these diseases from different angles, and we try to work in a nice group. Everyone has different knowledge and background, and but these are the main two diseases, dementia diseases we work on.
Wonderful, thank you. That's really good. So Mark, I'm going to come to you, and this might not be one that you're able to answer, but I wonder, with your kind of role looking over the whole of the Thames Valley network, so we've had somebody asking about research into diet, especially gluten excluding ketogenic diets. So it sounds like quite a specific dietary pattern, but do you know of any researchers in the network that are working on diet, or is it something that maybe other networks and centres are looking into?
So yes, so there's not that much research going on specifically in the network. There's some research looking at imaging, so brain imaging studies, and linking obviously cardiovascular health to brain imaging, and that brings in sort of dietary intake, so it kind of monitors the dietary intake, but it's not specifically geared up to assess whether those particular types of diets are going to slow down or be preventative. I think that the evidence around kind of dietary and dementia is very mixed. I guess knowing that a lot of kind of risk factors relate to cardiovascular disease, knowing that in essence that sort of Mediterranean diet can help cardiovascular health, that's obviously going to be beneficial for, in essence, brain health and cognitive function. But I guess the kind of categoric evidence is lacking, and that might just be the way that the trials are designed. So keep eating the fish and healthy foods, and hopefully that'll keep a healthy brain.
Wonderful, thank you. Good brain health message coming from you there, Mark. So sticking with brains, I guess the whole event is about brains, but Nora, you were showing some of the research that you do, which uses donated brain tissue, but also some of your colleagues use different methods. They use stem cells. So do these different methods, have they come together to give you a better understanding, and you kind of work collaboratively in that way to kind of build the knowledge about dementia?
Absolutely. We really need to have all the models together, because they each bring a little piece of the puzzle. So if you don't evaluate the postmortem human tissue that is really kindly donated by patients and their families, we don't know what's going on in the diseased brain. And this gives us clues, but because it's a diseased brain, it's only a snapshot. It's only the one picture we get out of the lifetime of a person. So it's quite limited. And for that, what we need to do is to then go back to the lab and use models where we can manipulate things, because that's the way science works. I put something in and have an observation. I take something out and have an observation. And in that way, we have learned a lot from mouse models, but there's always a difference between human and mice, human and a mouse, obviously. And that's why we've also brought in these iPSC, or induced pluripotent stem cell derived neurons, because they're great models of neurons. For example, in the case of Parkinson's, dopaminergic, or in the case of Alzheimer's, cortical models, where we can actually do these manipulations. The other thing is that we do still need the animals, because say, for example, the question that was just raised about diet, there's not a lot that I can do in the ways of manipulation of diet when it comes to a culture of a neuron in a lab, because our brains are designed, if everything you ate immediately was transduced to your brain, you could see the neurons in a wild shock of a sugar craze or in a wild shock of a lot of fat dancing around. And that's not how it works. There's a blood-brain barrier and they're very protected in their environment. And whilst we can do manipulations, they're not systemic in the way that we as humans will perceive our changes. And so we still need animal models. We need the full picture. We just can't have one or the other and actually produce the full package that we're going to need to find a disease modifying therapy.
Great, thank you. And thinking, I guess, along the process that research follows, so probably this question for you, Franziska. So your group is trying to find these new molecules that could be potential treatments. How long does it take to kind of find one of these potential, let's call them chemicals at this point, they're chemicals at that point, but then, and then what happens after your group has done their work? Who takes on that work, and how long could it be if it was successful to then reach people with that form of dementia?
That's a good question. It varies. You can't really say how much time it takes, but so the workflow is basically, we have a huge compound library we obtain from pharma companies or somewhere else, and we screen this library. We have a target protein which is involved in dementia, and then we screen this target against all these compounds in our compound library, and we get hits out of it. This means potential compounds that could hit our target and could be useful. So we filter this down, we funnel compounds through until we have only a few left. And these are the compounds we profile and characterize with all these with all the equipment we showed earlier in the video. And then whenever we have a reasonable amount of hits, good compounds, they will be taken further, and they will be, for example, given to Nora's lab, and they use these compounds to put them on their cells. And the amazing thing is that they not only have normal cells, they have stem cells, they have neurons. So yeah, this is like a nice chain, but it can vary. It could be quick, it could take years, but this is the really nice thing here in Oxford, for example. We have this very tight collaboration. So compounds will be given to the Oxford Parkinson's Disease Center, for example, where Nora is located. Yeah, it goes so much quicker because we have such a big group of expertise in the whole network, Reading and Oxford. Yeah, but there is no time range, you can tell, but it goes much quicker in this big team, at least. But yeah, this is the workflow we work with. Like a chain, basically, we hand over the compounds whenever we characterize them, and then it will get into in vitro on the cells, and then it will go in vivo in mice, and then it will go into patient cohorts finally. Yeah, but it's a matter of years. Yeah.
Wonderful. Yeah, I think I've seen estimates that it's somewhere between 10 or more years from finding that first chemical to then it potentially being, prescribed by a doctor to a patient. But I think for dementia, we've seen that a bit longer because the trials are longer, because the diseases are that bit more complicated. But yeah, so you're at that first stage, identifying the chemicals, finding the really good options that will then follow that pipeline through for further study.
Exactly.
Wonderful. We've had a couple of questions ahead of time about taking part in research, and I know that all of you work in the lab, but I wondered if anybody wanted to say anything about how, if people are involved with getting involved, if people are interested in getting involved with research studies, if there is a way to do that. I don't know, Mark, did you want to speak about that one?
Yeah, I think I can come in. Yeah, while we kind of mainly hide behind kind of lab doors and wear white coats, there is the kind of Joint Dementia Research Initiative. So hopefully, I'm sure some of our colleagues could share a link to that to our attendees to sign up. But I guess there is another way, is that you can come and visit us in the laboratory and share your experience of living with dementia and get in touch with us. And that, in essence, actually then produces new tangible outcomes. So one of the kind of evidences from us, we had people living with dementia come on site to visit the scientists here at the University of Reading. And from that stemmed a research grant to find out a little bit more about medicine use in dementia. So again, just by having conversations with people living with dementia, us as scientists can actually start to unpick what are the real challenges that these people are facing on a daily basis, and if science can resolve some of those issues that they have.
Brilliant, thank you. So yeah, I think we've shared the Join Dementia Research link in the chat, and that is a way to kind of indicate that you're interested in getting involved with research studies. So do check that one out. So I think we haven't got too many more questions coming through. So I'm going to hold on. I'm going to put a question to everyone. But if anyone in the audience does have a question, do put them in the Q&A so that we don't miss out asking them. But my question to each of you is, for you, or this might be your own research area, what is the most promising area of dementia research as far as you're concerned? So I will come to you first, Francesco.
Yeah, no, sorry, before I answer to that, I wanted to follow up a second on Mark's remarks. As a preclinical scientist who has been working always in the lab, I can only echo that, and I absolutely support that. It is fundamental, at least in my experience, been fundamental to have direct interaction with people with a lived experience of dementia, because the risk for people who works at a preclinical level is to have a view of dementia that comes entirely from papers. And while papers are scientifically accurate, they may lack that, further direct experience and connection, and for us to organize our work, I think is, I don't know, I think is great. It's been fundamental for me. So yes, that. Also, can I ask you to repeat the question, because I forgot?
What, from your perspective, is the most exciting or promising area of dementia research?
The most promising. Yeah, there are many. I mean, probably this is going to be a bit unpopular, but yeah, I do like the idea of ketogenic diet, not because of all the anecdotal knowledge. I mean, if you read on the internet, it seems that ketogenic diet cures everything, right? Although I must say that the only clinically accepted use for ketogenic diet, which I am aware of, is for the treatment of child epilepsy. Look at that. And we did observe indeed that, there is hyperexcitability in the brain of people with Alzheimer's disease. So there is some ground to explore that at a mechanistic level. Of course, it's very complicated because, as we said, there is a lot of stratification. So we may need to choose, people based on that. Beyond these, I think that at a preclinical level, I think, tau pathology now is raising a lot because amyloid pathology has been tackled down with some success, with some failures, with some controversy because of the brains packed with amyloid beta plaques of people that are perfectly functional at a cognitive level, which doesn't mean that they wouldn't have developed dementia anyways if they lived longer. So I think that now tau is a good one because it's clearer what sort of downstream signaling alterations are going on, and hence functional and hence cognitive. So personally, I'm focusing a lot on tau, more than I used to do in the past, and so I think there's a lot going on there. And then also on those, and we shouldn't forget that, on those clinical approaches which are aimed at bettering the quality of life of people with dementia, because I think there is a way to not, to reduce the impact of dementia at the very least. And so I think also that should be something we care about at a more multidisciplinary and interdisciplinary level.
Brilliant, thank you. That was a fantastic answer. So who should I come to next? Franziska, do you want to talk about what you think is the most promising area of research?
Yes, so spontaneously, I would say early Alzheimer's detection is there's a lot of effort going on at the moment. For example, detecting the disease having algorithms, when you can see in brain scans very early on that there's an accumulation happening and early detection of the disease. I think this is quite promising at the moment because the technology advances a lot, resolutions of scan equipment is advancing, and this is the one point I wanted to raise. So there's definitely a lot of effort going in at the moment.
Brilliant, thank you. Yes, picking up early, way before symptoms appear, would be really, really valuable. So next, Nora, can I come to you? What do you think is the most promising area of research?
Of course, I'm torn between a mixture of what Francesco and Francisca said, actually. On the one side, I think that early biomarker detection is a must. We really need really anything that we can screen the population with and detect it soon. So perhaps scans are not what I'm thinking of. It's probably more in the periphery or in blood samples, something that we can really just have a glance at the whole population and define whether people are at risk or not, because we're really lacking those. And the other one is back to Francesco, aggregation, really, not specifically to tau in my case. I study aggregation of synuclein in the case of Parkinson's and tau in the case of Alzheimer's, but it's really common in basically any neurodegenerative condition. And I think they go hand in hand, because the thing is that we know that normally aggregation just goes on and on and on and accumulates through disease. But if we don't have that biomarker window, if we don't have that access to the information, we don't know where each patient is, and we don't know what we need to treat them for. And it's really possible that we don't need to treat the same people that have been living with disease for a year or 10 years. It's mind-boggling that we would, right? And it's again what Francesco was saying earlier about disease stratification and like building groups of patients and really looking at the different pathologies that there are between our patients and tackling everything when and where we need to.
Brilliant. Yes, that kind of concept of personalized medicine, understanding what's happening with that person so that you're giving them the best options and tailoring the treatments to them. Yeah, that would be really great. And finally, coming to you, Mark, what do you think is the most promising area of dementia research?
Yes, I kind of share and echo the sentiments raised by the other researchers, but I'm going to be a little bit different for my area of research. And just thinking about the support cells, those glial cells. I think there's a lot of information coming out now in the laboratory about how these cells change and indeed can afford protection against the neurons. So we know that the brain cells, the neurons, are the ones that are susceptible to death, and those are the ones that we lose as the disease progresses. But how about these other cells that make up our brain? And I think efforts to target them, because they're a little bit more robust and resilient, we might be able to modify their activity easier than, in essence, targeting the neuron. So watch this space. I think there's a lot more information to come about these support cells and how they could combat dementia.
Brilliant, thank you. So just as we're about to wrap up, I can see we've had a couple of questions come through, but I think they're more related to kind of clinical aspects. And unfortunately, our speakers today are all lab-based scientists, so we haven't really got anyone with the expertise to answer those, but we will point you in the direction of somewhere where you can probably get those questions answered. So just hold on and we'll tell you where to go with those questions. So I just want to now, as I'm wrapping up, say a huge thank you to Mark, Nora, Francesco and Francisca for coming along today, for coordinating those wonderful films. It was fantastic to see behind the scenes of the lab and the amazing work that you're doing. So thank you, and I will say goodbye to you now before I then briefly wrap up for everyone else.
So it's goodbye from me. I hope you've enjoyed this afternoon's session as much as we did in putting it together. So goodbye.
Thank you very much for the invite today. It's been, oh, sorry, I pushed the wrong button. Thank you very much today. It's coming. Yes, I had to make a mistake before the end of it. It's been great talking about my my research and meeting you all, even though virtually. And I hope to be able to see you soon in person.
Yeah, thank you very much for your interest and for watching the videos. We had a lot of fun making this. And yeah, I hope you took a lot of information out of this event, and I wish you a very good afternoon.
And lastly, also from me, thank you so much for allowing us to share what we do in the lab with you, and for your interest, and we hope to see you soon.
Brilliant. So thank you very much to our speakers. If you as the audience could stick with me just a couple of minutes longer, we just want to do a closing poll to try and understand how you found today and get some insight into how it's all gone. So if we can get the poll questions up on screen. So it's, would you recommend these events to your friends and family? Did you learn anything new about dementia research today? And would you attend future Lab Notes events? So I'll pause for a second while you can pop your answers in. So hopefully that's given you enough time to just answer those, and we can have a little look at the results. So you would recommend the events to friends and family, you have learned something, and you would attend again. That's wonderful, and really helps support why we do these, and helps us to continue to shape these for the future. So thank you so much for coming along. We'll be sending a feedback survey along with the video recording of this event next week. So please do complete the survey, as we'd appreciate any of the more detailed feedback you can give us about how this has gone. And we can continue to improve. The next event in the series is on the 1st of July at 7 p.m. and features researchers from our Wells Research Network. They will be sharing their research studying particular genes and how they affect brain biology in dementia. It will be a chance to hear about how the work that follows the genetic discovery is taking place, and how this can lead to potential new treatments being discovered. So do come along in a few weeks' time for what is set to be another great event. You can find out more and sign up on our website to join us on the day. I know there were some questions that we weren't able to answer today, and some where you may need more personalised information or guidance on where to turn. Our dementia research info line is here to help. They can answer questions and signpost to other sources of information and support. We also spoke a little bit about taking part in research, so just another reminder about joint dementia research, and our dementia research info line can help provide more information about that and help you sign up. So that is it from me today, and I just want to say a huge thank you for coming along, for all your questions, and I hope you found it really interesting and useful, and will join us again for another event in our series. So thank you and goodbye.